Welcome to Chapter 9, Section 5, How Genes Are Regulated. By the end of this section, you'll be able to discuss why every cell does not express all of its genes, describe how prokaryotic gene expression occurs at the transcriptional level, understand that eukaryotic gene expression occurs at the epigenetic, transcriptional, post-transcriptional, translational, and post-translational levels. For a cell to function properly, necessary proteins must be synthesized at the proper time. All organisms and cells control or regulate transcription and translation of their DNA into proteins. The process of turning on a gene to produce RNA and protein is called gene expression. Whether in a simple unicellular organism or in a complex multicellular organism, each cell controls when and how its genes are expressed. For this to occur, there must be a mechanism to control when a gene is expressed to make RNA and protein, how much of the protein is made, and when it is time to stop making that protein because it is no longer needed. Cells in multicellular organisms are specialized. Cells in different tissues look very different and perform different functions. For example, a muscle cell is very different from a liver cell, which is very different from a skin cell. These differences are a consequence of the expression of different sets of genes in each of these cells. All cells have certain basic functions they must perform for themselves, such as converting the energy in sugar molecules into energy in ATP. Each cell also has many genes that are not expressed, and expresses many that are not expressed by other cells, such that it can carry out its specialized functions. In addition, cells will turn on or off certain genes at different times in response to changes in the environment or at different times during the development of the organism. Unicellular organisms, both eukaryotic and prokaryotic, also turn on and off genes in response to the demands of their environment so that they can respond to special conditions. The control of gene expression is extremely complex. Malfunctions in this process are detrimental to the cell and can lead to the development of many diseases, including cancer. Prokaryotic versus eukaryotic gene expression. To understand how gene expression is regulated, we must first understand how a gene becomes a functional protein in a cell. This process occurs in both prokaryotic and eukaryotic cells, just in slightly different fashions. Because prokaryotic organisms lack a cell nucleus, the process of transcription and translation occur almost simultaneously. When the protein is no longer needed, transcription stops. As a result, the primary method to control what type and how much protein is expressed in a prokaryotic cell is through the regulation of DNA transcription into RNA. All these subsequent steps happen automatically. When more protein is required, more transcription occurs. Therefore, in prokaryotic cells, the control of gene expression is almost entirely at the transcriptional level. The first example of such control was discovered using E. coli in the 1950s and 1960s by French researchers and is called the Lac Operon. The Lac Operon is a stretch of DNA with 3 adjacent genes that code for proteins that participate in the absorption and metabolism of lactose, the disaccharide, a food source for E. coli. When lactose is not present in the bacterium's environment, the Lac genes are transcribed in small amounts. When lactose is present, the genes are transcribed and the bacterium is able to use the lactose as a food source. Okay, when there's no glucose. Glucose is a preferred food source, and that's a different aspect of regulation of the Lac Operon. If you are out of glucose, then you would definitely want your alternative food source in the form of lactose to be able to be readily broken apart. But if you have plenty of glucose, you'd rather eat the glucose. The operon also contains a promoter sequence to which the RNA polymerase binds to begin transcription. Between the promoter and the 3 genes is a region called the operator. When there is no lactose present, a protein known as repressor binds to the operator and prevents RNA polymerase from binding to the promoter, except in rare cases. Thus, very little of the protein products of the 3 genes is made. When lactose is present, an end product of lactose metabolism binds to the repressor protein and prevents it from binding to the operator. This allows RNA polymerase to bind to the promoter and freely transcribe the 3 genes, allowing the organism to metabolize the lactose. And again, this is largely in the absence of glucose. Eukaryotic cells, in contrast, have intracellular organelles and are much more complex. Recall that in eukaryotic cells, the DNA is contained inside the cell's nucleus, and it is transcribed into messenger RNA there. The newly synthesized mRNA is then transported out of the nucleus into the cytoplasm, where ribosomes translate the mRNA into protein. The process of transcription and translation are physically separated by the nuclear membrane. Transcription occurs only within the nucleus, and translation only occurs outside the nucleus in the cytoplasm. The regulation of gene expression can occur at all stages of the process. Regulation may occur when the DNA is uncoiled and loosened from the nucleosomes to bind transcription factors, epigenetic level, when the RNA is transcribed, transcriptional level, when RNA is processed and exported to the cytoplasm after it is transcribed, post-transcriptional level, when the RNA is translated into protein, translational level, or after the protein has been made, post-translational level. The differences in the regulation of gene expression between prokaryotes and eukaryotes are summarized in this table. In the prokaryotic organisms, we don't have a nucleus, but we do in the eukaryotes. RNA transcription and protein translation occur almost simultaneously in prokaryotes, but they are separated in eukaryotes. Gene expression is regulated primarily at the transcriptional level in prokaryotes, but can occur at any level, any of those levels mentioned, in eukaryotes. Evolution in action, alternative RNA splicing. In the 1970s, genes were first observed that exhibited alternative RNA splicing. Alternative RNA splicing is a mechanism that allows different protein products to be produced from one gene when different combinations of introns, and sometimes exons, are removed from the transcript. This alternative splicing can be haphazard, but more often it is controlled and acts as a mechanism of gene regulation. With the frequency of different splicing alternatives controlled by the cell as a way to control the production of different protein products in different cells, or at different stages of development. Alternative splicing is now understood to be a common mechanism of gene regulation in eukaryotes. According to one estimate, 70% of genes in humans are expressed as multiple proteins through alternative splicing. How could alternative splicing evolve? Introns have a beginning and an ending recognition sequence, and it is easy to imagine the failure of the splicing mechanism to identify the end of an intron and find the end of the next intron, thus removing 2 introns and the intervening exon. In fact, there are mechanisms in place to prevent such exon skipping, but mutations are likely to lead to their failure. Such mistakes would more than likely produce a non-functional protein. Indeed, the cause of many genetic diseases is alternative splicing rather than mutations in a sequence. However, alternative splicing would create a protein variant without the loss of the original protein, opening up possibilities for adaptation of the new variant to new functions. Gene duplication has played an important role in the evolution of new functions in a similar way, by providing genes that may evolve without eliminating the original functional protein. And that brings us to the end of Chapter 9. Join me next time for Chapter 10, biotechnology. Until then,